Let us study viruses. Viruses are very small, acellular, and obligate intracellular particles that infect and take over a host cell in order to replicate. Viruses are microscopic infectious agents that replicate only inside the living cells of all organisms because they lack chemical machinery for generating energy and to synthesize their own large molecules. So, to replicate themselves, they must find appropriate host cells. Therefore, they often cause disease. So, viruses are not considered as living organisms because they lack the cellular structure and metabolic processes necessary for life. Since most viruses are ultra microscopic, they can be seen under electron microscope. Let us see the characteristics of viruses. Viruses have inner core of nucleic acid that is surrounded by protein coats. Viruses cannot be grown on artificial cell-free media, but grow in cells of animals, plants, bacteria, protozoa, algae, and fungi. That means they grow in all living cells since they are obligate intracellular parasites. Viruses do not have cellular organizations like cell wall, cell membrane, and cellular organelles, even ribosomes. They do not occur free in nature. Molecules on virus surface impart high specificity for attachment to host cells. The reason why most viruses are host specific. Viruses are not affected by antibiotics because antibiotics are specifically designed to target bacteria, not viruses. Antibiotics work by attacking bacterial structures or processes like cell walls, protein synthesis, or DNA replication, which viruses do not have. Viruses are inert or nucleoprotein filterable agents when they are outside a host cell. This means they cannot move, grow, reproduce, or carry out metabolic processes on their own. Viruses occupy a space in between living and non-living because they are inactive macromolecules outside the host cells and only active inside host cells. When we see the sizes of viruses, they are ultra microscopic. That means they range from 20 nanometer to 450 nanometer in diameter. Viruses do not independently fulfill the characteristics of life. They have basic structure that consists of protein shell, what we call capsid, surrounding nucleic acid core. Nucleic acid of the viral genome is either RNA or DNA, but not both. When we see the genetic materials of viruses nucleic acid can be double-stranded DNA, single-stranded DNA, single-stranded RNA, or double-stranded RNA. Viruses multiply by taking control of host cells' genetic material or machinery to regulate their synthesis, assembly of new viral particles because viruses lack machinery or enzymes necessary for synthesis of their protein and genetic materials. Let us see basic structural components of viruses. Basic structure of viruses include nucleic acid core or either DNA or RNA, that is the genome of viruses. The second structural component is the surrounding protein coat, what we call capsid, which is made from capsomers. These are repeated identical protein subunits to make capsid. Nucleocapsid is a combination of protein and the core or genetic material. The third component is enzymes or accessory proteins. This present in some viruses, not in all viruses. This play a role during infection For example, lysozymes make hole in bacteria cell to let in viral nucleic acids enter. Another enzymes or accessory proteins, nucleic acid polymerase. This is very important to transcribe viral genome into mRNA during replication process. The third enzyme, reverse transcriptase, to convert viral RNA to viral DNA. This is RNA-dependent DNA polymerase present in retroviruses. The fourth component includes envelope, which is present in some viruses. This is made from glycoprotein and phospholipid bilayer, and this envelope present outside capsid proteins. The fifth one is matrix protein, which is used to connect capsid with envelope. The sixth component includes spikes or glycoproteins that protrude out the envelope, which might be used by viruses for attachment purpose. That means to attach themselves on the host. Viral symmetry, classification of viruses based on their structures. Viral symmetry refers to the structural arrangement of proteins in the capsid that surround virus's genetic material. Let us see helical symmetry. The capsid proteins wrap around the viral genome in a spiral or helical arrangement, forming a rod-like or filamentous shape. There are several viruses found with helical morphology. They consist of identical protein subunits or protomers or capsomers, which is assembled in helical structure around the viral genome. The protein subunits generally form a rigid nucleocapsid. The helical structure provides flexibility to the filaments. The best example here is tobacco-mosaic virus and Sendai virus. The second type of viral symmetry is icosahedral symmetry. This refers to a type of polyhedron with 20 equilateral triangular faces, 12 vertices or corners, and 30 edges. The rigid structure provides protection to the genome. The common example here is papovavirus, picornavirus, adenovirus, and Toga virus. The third viral symmetry is complex symmetry. Complex symmetry of viruses are viruses with structures that do not fit neatly into the regular geometric classification of icosahedral or helical symmetry. These viruses have intricate designs combining multiple structural features. Common example is pox virus. So generally, viruses occur in three main shapes: spherical or polyhedral, cylindrical or helical, and complex structures. Spherical or polyhedral can be naked capsid or non-enveloped and enveloped. Naked capsid includes poliovirus, adenovirus, and papillomavirus. Enveloped virus include herpes simplex virus. Cylindrical or helical virus can be non-enveloped or naked capsid and enveloped. Naked capsid or non-enveloped virus include bacteriophage M13 and tobacco mosaic virus. Enveloped virus include influenza virus. Classification of viruses. Today, the primary criteria for delineating main viral taxa are: the first one, type and character of the viral genome and their strategy of replication. In this case, they can be DNA virus, RNA virus, and retroviruses. Based on the type of organism they infect or based on their host organisms, they can be animal infecting, plant infecting, and bacteria infecting or bacteriophage. Even though viruses infect all life forms in this world, classification of viruses based on the nature of their genetic material and the way how they replicate or express themselves. The first one is DNA virus. DNA virus is a group of viruses that have DNA as a genetic material. When we see their ways of expression, first, the virus attaches itself to the host cell. Second, after it attaches on the host cell, it injects or relays their DNA into the host cell. The third step, the genetic material DNA can replicate itself and can control viral protein synthesis. Fourth, the new viral DNA and new viral protein assembled to form new viral particles. And lastly, the new viral particles leave the host cell. The best example is herpes simplex, smallpox, and hepatitis B. The third group is RNA virus. This is a group of viruses that have RNA as a genetic material. When we see their ways of expression, the virus must attach itself to the host cell. Second, the virus relays or injects its RNA into the host cell. Third, RNA can be used to synthesize more viral proteins or enzymes. Fourth, the synthesized enzymes here used to control the production of more viral RNA. Fifth, the new RNA and new protein assembled to new viral particles. Six, the new viral particles leave the host cell. The best example here is poliomyelitis and H1N1 virus. The third group is retrovirus. This is a group of viruses that have RNA as a genetic material which can be reversibly transcribed into DNA once the genetic material enters into the host's cytoplasm. When we see their ways of expression, the virus attach to the host cell. Second, the virus inject or relays their RNA and enzymes reverse transcriptase into the host cell. Third, their genetic material RNA reversely transcribed to DNA via reverse transcriptase enzyme. Fourth, viral DNA then enter into the host nucleus and integrate with the host genome. Fifth, viral DNA controls the formation of more viral proteins and RNA. Then, new viral RNA and new viral protein assembled to produce new viral particles. The new viral particles are then leave the host cell. Generally, the difference between DNA, RNA, and retroviruses are DNA viruses contain DNA as their genetic material. DNA viruses mostly contain double-stranded DNA molecule. DNA viruses have less mutation rate and they replicate inside the host nucleus. RNA viruses contain RNA as their genetic material, and this genetic material RNA is single-stranded. RNA viruses have high mutation rate since they contain RNA as their genetic material and they replicate in the cytoplasm of the host cell. Provirus, a unique group of virus that has RNA as a genetic material and convert its genetic information from RNA into DNA after the virus has infected a host cell or a host organism. When we see viral replication, viruses invade all living host cells that are 1,000 or more times bigger than viral size. In replication processes, viruses hijack the metabolism of host cells to produce many viral copies. After viruses make their copies, they often destroy the host cell when the new viral particles leave or release from the host cell. Viral replication has been studied in a wide range of viruses and their host cells. One of the best studied processes of replication is that carried out by bacteriophages of the T-even group, like bacteriophage T2, bacteriophage T4, bacteriophage T6, and 8. As I told you before, in general, viruses go through the following five main steps in their replication cycles to produce more virions. The first step here is adsorption. This is the attachment of viruses to the host cell. The second step here is penetration. That is the entry of virions or viral genome into host cells. The third step, synthesis. That means synthesis of new viral nucleic acid, viral protein, that might be capsid proteins, and other viral components. The fourth step is maturation. This is the assembly of newly synthesized viral components into new complete virions. The fifth step here is release. This is the departure of new virions from the host cells. So in this case, the virus may kill the host cells through lysis, but not always. Bacteriophage. Bacteriophages are bacterial viruses that infect bacteria. These are obligate intracellular parasites that multiply inside bacteria by making use of some or all of the host biosynthesis machineries and resources. This bacterial infecting virus is called phages. This is hexagonal head of bacteriophage that is determined by capsid protein to enclose DNA core or genome of the virus. This is the genome of bacteriophage, which carries the genetic information necessary for replication of new phage particles. This is the tail chest part that retracts or contracts to move the genome from the head into the host cell's cytoplasm. This is the base plate, prongs or pin, and tail fibers that attach phage to specific receptor sites on the cell wall of a specific susceptible host bacteria. Phages or viruses exhibit two different types of life cycle, that is, lytic cycle and lysogenic cycle. The first one is lytic cycle or sometimes called virulent cycle. This is an intracellular multiplication of the phage or viruses that result in the lysis of host cell or host bacteria and release of progeny virions or viral particles. Lysozyme in this case, which is coded by phage gene, break down the cell wall and allowing the new viruses to escape the host cell. Therefore, phages or viruses such as T4 are called virulent or lytic phage because they lyse and destroy the bacteria or host cell they infect. Such infection by virulent phages is lytic cycle of infection. The released phages can now infect more susceptible bacteria and starting the infection process all over again. As we can see from the diagram, in case of lytic cycle, first the virus or phage attach itself on the host cell surface or receptors. Second, the virus inject its genetic material into the host cytoplasm, then degrade host DNA and control early protein synthesis, that is, phage or viral enzyme synthesis. Third, phage or viral DNA replicate and hence determine synthesis of late structural proteins, what we call capsid proteins. Fourth, the viral DNA and proteins are assembled together. And lastly, release of new phage particles through lysis of host cell. The second form of viral life cycle is lysogenic cycle. Lysogenic cycle is a phase in the life cycle of certain viruses, particularly temperate bacteriophages like lambdaphage, during which the viral DNA integrates into the host cell's genome and remains dormant as a prophage. Unlike the lytic cycle, The lysogenic cycle does not immediately destroy the host cell, but instead allows the virus to coexist with the host. That means, unlike virulent phages, some phages, what we call temperate phages, integrate into the genome of the bacterial chromosome without causing any lysis of the bacteria. The integrated phage nucleic acid is called prophage. The prophage behaves like a segment of the host chromosome and multiplies synchronously with the host chromosome. This phenomena is known as lysogeny. The bacterium that carries a prophage within its genome is called lysogenic bacterium or lysogenic host, and the phages are called lysogenic or temperate phages. As we can see from the diagram, in a lysogenic cycle, the first step here is attachment or adsorption. In this case, the bacteriophage attaches to the surface of the host cell using specific receptor proteins. The second stage here is entry or penetration. In this case, the viral DNA or RNA is injected into the host cell by leaving the protein coat outside the host cell. The third step is integration. The viral genome integrates into the host DNA by forming a prophage. This process is mediated by viral enzymes like integrase enzyme. That means phage DNA inserts itself as a prophage into bacterial chromosome. The fourth step is latency. The prophage remains dormant and is replicated along with the host's DNA during cell division. That means prophage is replicated along with the bacterial DNA prior to or before binary fission or cell division. The fifth step is propagation. Each time the host cells divide indefinitely, another step is induction or switch to lytic cycle. When triggered by stress factors or environmental factors like UV light, chemical agents, or DNA damage, the prophage exercises itself from the host genome. Then the virus enters the lytic cycle and leading to active replication, assembly of new virions and cell lysis. That means the phage DNA directs the cell's metabolism to produce viral components like proteins and copies of phage DNA. Certain protein molecules assemble and produce empty phage heads. Phage heads and pieces of DNA molecule assembled together. Then collars, chassis, and base plates have been attached to heads. Tail fibers are added lastly. Then lastly, bacterial cell lysis to release completed infective phages. Okay, let's get compare lytic cycle with lysogenic cycle. In lytic cycle, the DNA of the virus does not integrate into the host DNA, but in lysogenic cycle, the DNA of the virus integrates into the host DNA. In lytic cycle, host DNA is hydrolyzed, but in lysogenic cycle, host DNA not hydrolyzed. In lytic cycle, there is absence of prophage stage, but in lysogenic cycle, presence of prophage stage. In lytic cycle, DNA replication of virus takes place independently from the host DNA replication. But in lysogenic cycle, DNA replication of the virus takes place along with the host DNA replication. Lytic cycle occurs within a short period of time. Lysogenic cycle takes time. In lytic cycle, symptoms of viral replication are evident, but in lysogenic cycle, symptoms of viral replication is not evident. In lytic cycle, genetic recombination in the host bacterium not allowed, but in lysogenic cycle, genetic recombination in the host bacterium is allowed. In lytic cycle, the cellular mechanism of the host cell is totally undertaken by viral genome. But in lysogenic cycle, the cellular mechanism of the host cell is somewhat disturbed by viral genome.